Welcome to this biology quiz on mitosis. In this video, we will answer and explain 15 important multiple-choice questions about mitosis. Let's begin. Question 1. What is the main purpose of mitosis in multicellular organisms? A. Production of gametes. B. Growth and tissue repair. C. Reduction of chromosome number. D. Genetic variation. The correct answer is B. Growth and tissue repair. Mitosis produces identical body cells that help organisms grow, replace old cells, and repair damaged tissues. It does not produce gametes. Gametes are produced by meiosis. Question 2. In which phase of mitosis do chromosomes line up at the equator of the cell? A. Prophase. B. Metaphase. C. Anaphase. D. Telophase. The correct answer is B. Metaphase. During metaphase, chromosomes arrange themselves along the center or equator of a cell. This alignment helps ensure that each daughter cell receives an equal set of chromosomes. Question 3. During which stage do sister chromatids separate and move to opposite poles? A. Prophase. B. Metaphase. C. Anaphase. D. Cytokinesis. The correct answer is C. Anaphase. In anaphase, spindle fibers pull sister chromatids apart toward opposite poles of the cell. This is one of the most important events in mitosis. Question 4. What structure helps move chromosomes during mitosis? A. Ribosomes. B. Spindle fibers. C. Golgi apparatus. D. Vacuole. The correct answer is B. Spindle fibers. Spindle fibers attach to chromosomes and help move them during cell division. Without spindle fibers, chromosomes could not separate properly. Question 5. Which event occurs during prophase? A. Nuclear membrane reforms. B. Cytoplasm divides. C. Chromatids separate. D. Chromosomes condense and become visible. The correct answer is D. Chromosomes condense and become visible. During prophase, chromatin coils tightly into visible chromosomes. The nuclear membrane also begins to break down. Question 6. A scientist observes a cell with chromosomes aligned in the middle of the cell. Which stage is being observed and why? A. Metaphase. Chromosomes align at the equator. B. Prophase. Chromosomes are condensing. C. Anaphase. Chromatids are separating. D. Telophase. Nuclei are reforming. The correct answer is Metaphase. Chromosomes align at the equator. The key clue is that the chromosomes are arranged in the middle of the cell. That is the defining feature of metaphase. Question 7. If spindle fibers fail to form during mitosis, what is the most likely result? A. DNA replication increases. B. Chromosomes cannot move properly. C. Cytoplasm divides faster. D. The nucleus disappears permanently. The correct answer is B. Chromosomes cannot move properly. Spindle fibers are responsible for moving chromosomes. Without them, chromosomes may not separate correctly, leading to abnormal daughter cells. Question 8. Why is mitosis considered important for maintaining genetic stability? A. It produces genetically different daughter cells. B. Chromosome number is halved. C. Daughter cells receive identical chromosomes. D. It causes mutations intentionally. The correct answer is C. Daughter cells receive identical chromosomes. Mitosis ensures that each daughter cell gets the same genetic information as the parent cell. This maintains stability in the organism. Question 9. A skin wound heals after several days because cells undergo mitosis. Which statement best explains this? A. Mitosis reduces chromosome number. B. Mitosis creates identical replacement cells. C. Mitosis forms gametes for repair. D. Mitosis changes cell functions completely. The correct answer is B. Mitosis creates identical replacement cells. When skin cells are damaged, mitosis produces new cells that replace the injured tissue. These new cells are genetically identical to the original cells. Question 10. Cancer is often linked to uncontrolled mitosis. What does this mean? A. Cells stop dividing completely. B. Cells divide without regulation. C. Chromosomes disappear. D. Cells become smaller only. The correct answer is B. Cells divide without regulation. Normally, cell division is carefully controlled. In cancer, cells continue dividing uncontrollably, forming tumors and damaging tissues. Question 11. A cell contains 12 chromosomes before mitosis. How many chromosomes will each daughter cell contain after mitosis? A. 12. B. 6. C. 24. D. 48. The correct answer is A. 12. Mitosis produces daughter cells with the same number of chromosomes as the parent cell. So if the original cell has 12 chromosomes, each daughter cell will also have 12. Question 12. A student observes two new nuclei forming at opposite poles of the cell. Which stage is this? A. Metaphase. B. Anaphase. C. Telophase. D. Cytokinesis. The correct answer is C. Telophase. During telophase, nuclear membranes reform around the chromosomes at opposite poles, creating two new nuclei. Question 13. Question 13. Which sequence correctly shows the stages of mitosis? A. Prophase, metaphase, anaphase, telophase. B. Metaphase, prophase, telophase, anaphase. C. Anaphase, metaphase, prophase, telophase. D. Telophase, anaphase, metaphase, prophase. The correct answer is A. Prophase, metaphase, anaphase, telophase. A simple way to remember this order is PMAT, prophase, metaphase, anaphase, and telophase. Question 14. A plant root tip has many cells undergoing mitosis. Why are root tips good regions for observing mitosis? A. Cells are divide rapidly for growth. B. Cells are never divide. C. Root cells lack chromosomes. D. Photosynthesis occurs there. The correct answer is A. Cells that divide rapidly for growth. Root tips are active growth regions, so many cells are continuously dividing by mitosis. Question 15. A cell in anaphase suddenly stops dividing before chromatids reach opposite poles. What problem could occur? A. Daughter cells may receive unequal chromosomes. B. DNA replication doubles again immediately. C. Cytoplasm disappears permanently. D. The cell becomes a gamete. The correct answer is A. Daughter cells may receive unequal chromosomes. If chromatids fail to separate properly, the daughter cells may receive incorrect chromosome numbers, leading to abnormalities.